Hi, I'm Lorely Peñaverde. My topic is the cell cycle regulation. I chose this topic because I usually teach the cell cycle lesson for middle school students, and I want to have a more in-depth understanding of the concepts and share the information with you. I hope you find my presentation interesting and helpful as well. For my presentation, I will be able to explain the role of cyclin and cyclin-dependent kinases in cell cycle regulations. I will discuss the function and importance of checkpoints in the cell cycle. Finally, I will describe how a dysfunction in the cell cycle can potentially cause cancer. Nelson and Cox 2017 mentioned that a cell's decision to whether divide or not divide is crucial to an organism's survival, since every organism undergoes cell division, primarily for reproduction or growth and development. The unicellular organism that multiplies, or a single human fertilized egg that eventually develops into an adult multicellular organism, may differentiate into various types and perform its function accordingly. Particularly, eukaryotic cells decide to divide in precise biological sequences regulated by signaling mechanisms that any problem affecting the cell cycle is detrimental to an organism. What happens during a cell cycle? There are so many events occurring during the cell cycle. Barnum O'Connell 2014 described the cell cycle as a series of events in which cellular components are doubled and then accurately segregated into the daughter cells. The cell has to undergo a cell cycle to perform these functions properly. The cell is so busy doing these processes all throughout the cell cycle that it might ask for a vacation. Moreover, it is important to control the processes of the cell cycle. Collins, Jacks, and Pavlitich 1997 explained that there are at least two types of cell cycle control mechanism. The first one is a cascade of protein phosphorylation that relay a cell cycle from one stage to the next, which means adding phosphoryl group to proteins. Then the second one is the set of checkpoints that monitor the completion of critical events and delay progression to the next stage if necessary. Personally, I find this description precisely explaining the mechanisms that I will discuss for this presentation. Again, the mechanisms are protein phosphorylation and dephosphorylation of some enzymes, as well as the checkpoints that determine whether the cell cycle delays or progress for its completion. But before we continue, first I will review the stages of the cell cycle. Then I will explain the role of the protein kinases and the checkpoints in the stages. So as illustrated, the phases of the cell cycle involve four stages. There are the S phase, or the synthesis phase, the G1 and the G2 phase, which means the gap between divisions, which are part of the interphase, and then this part is the mitosis, which consists of many stages itself. So we have the G2 phase here that shows that the G2 phase and the S phase. The S phase is the synthesis phase. The cell replicates its DNA to produce two copies that will be distributed for two daughter cells that are produced during cell division. The G2 phase, on the other hand, is the synthesis of new proteins occurring in this phase. The cells grow and double in size to prepare to undergo the mitosis. The hours right here, the 6 to 8 hours for synthesis phase, is approximate time the phase occurs in a cycle. So the G2 phase is about 3 to 4 hours. Now, if we go back after the synthesis and the G2 phase, we have the mitosis. So I will review briefly what are the stages of the mitosis again. So mitosis is the process of cell division, producing daughter cells from parent cells and distributing the genetic material to its daughter cells. So if you take a look, these are the phases of mitosis. So at the G2 phases, the cells have loosely coiled and replicated chromosomes. But at the beginning of the mitosis, it will start with a breakdown of the nuclear membrane. And the next phase, the prophase, it will have the spindle fibers that appear and the chromosomes will condense. Then at the prometaphase, the spindle fibers attach to the chromosomes and the chromosomes condense. Now, what do you think happens here in the metaphase? If you say that it's the chromosomes alignment, then you are correct. The next one is the centromeres divide and sister chromatids move to the opposite poles. When that happens, the nuclear membrane reforms and chromosomes decondense, and the spindle fibers will now appear. Now on the cytokinesis, on the last stage, we will have two new daughter cells from the parent cell as the cytoplasm divides, and they will have identical genetic information. Now, after the mitosis, we can see here that there is the G0 phase. The G0 phase, so we have the S phase that the synthesis happened or occurred. We have the G2 phase where the growth and doubling in size occurs. We have the mitosis where there's two new daughter cells with each genetic information. When the cycle reaches the G0 phase, the cell will then enter into the G1 phase, but in the G0 phase, the cell may continue or stop dividing, lasting from hours to lifetime of the cell. This according to Nelson and Cox, 2017. Accordingly, it may enter into a quiescent stage, wherein the cells rest and do not replicate before being reactivated or enter the cell again through the G1 phase. Now, the cells that already differentiated with specific function and form, such as neurons and adipocytes, remain in the G0 phase, whereas other cells, such as the stem cells that are undifferentiated cells that can mature into varying types of adult cells, proceed to the next stage. Also, cells can lose their ability to divide and grow due to aging or cellular senescence and experience apoptosis or cell death as well. Now for the G1 phase, right here, the G1 phase, the daughter cells prepare to divide again during the G1 phase, wherein RNA and protein synthesis occurs as the cell enters the synthesis phase of the cycle. The cell does not rest both in G1 and G2 phases. Instead, these are the periods where they obtain mass, integrate growth signals, organize a replicated genome, and prepare for chromosome segregation. That's according to Barnum and O'Connell of 2014. Now, if we take a look, right here, there are two checkpoints in the G1 and in the G2 stages. There's a restriction point or the checkpoint in which a cell that passes the restriction point can now pass. If it's from the G1, it can pass on the synthesis, and if it's from the G2, it can pass on the mitosis. Now, the cell checkpoints, according to Barnum and O'Connell, 2014, the cell checkpoints are surveillance mechanisms that monitor the order, integrity, and fidelity of the major events of the cell cycle, which include appropriate cell size, DNA replication, and accurate segregation to each daughter cell during mitosis. The cell cycle checkpoints identify the damages in the critical cell cycle events such as DNA replication and chromosome segregation. Correspondingly, the cell size checkpoints have been observed in phases G1 and G2, although some observations apparently show that the size of the daughter cells after mitosis affect the progression of the cell cycle. The varying types of cell cannot specifically distinguish how the cell size can affect the cycle entirely. Moreover, the checkpoints are vital to ensure that the cell does not attempt mitosis until the replication is complete, because the cells might have the risk of reduced ploidy, haploid, diploid, or the number of chromosomes for each daughter cells. The synthesis phase is an important phase for the cell to cope with the DNA damages after the checkpoint in G2 phase. That's according still from Barnum and O'Connell of 2014. Now, this orderly sequence of the cell cycle is regulated by a family of protein kinases. Through the process of phosphorylating proteins at specific time intervals, all of the cell metabolic activities needed to complete the cycle occurs accordingly. Cyclin is the regulatory unit, while the cyclin-dependent kinase, or CDK, is the catalytic subunit that binds together to catalyze the progression of the cell cycle. The CDKs are serine threonine protein kinases that phosphorylate key substrates to promote DNA synthesis and progression during mitosis. So we have here the CDK and the cyclin. They bind together, phosphorylated by protein. Then it will have activated CDK cyclin complex that will target the protein. Now, different organisms have different amounts of cyclins and CDKs. The animals have at least 10 different cycles that act with at least 8 CDKs in various combinations and checkpoints during the cell cycle. In this figure, Nelson and Cox presented that there are different activities in each phase in an animal cycle. If you notice, the cyclin E and CDK2 combination showed a peak between the G1 phase, right here, and the S phase boundary, the moment wherein enzyme synthesis is needed for DNA replication. On the other hand, the cyclin A CDK2 activity rises during the S and the G2 phases, then drops sharply in the M, while the cyclin B and CDK1 peaks. Correspondingly, there are mechanisms that regulate CDK activities. These are the phosphorylation or dephosphorylation of the CDK. The second one is the controlled degradation of the cyclin subunit, periodic synthesis of CDKs and cyclin, and the action of specific CDK inhibiting proteins. These processes attribute to cell oscillation, which is a type of cell signaling that controls each stage of the cell cycle, ensuring that each stage is completed before moving on to the next stage. I will use the illustration here to explain the first two mechanisms. The phosphorylation of THR160 or threonine 160 of CDK2 and the dephosphorylation of tyrosine 15 of CDK2 are both critical in regulating the activities of a CDK. The phosphorylation of THR160 opens the cyclin to bind to protein substrates, whereas the dephosphorylation of protein tyrosine 15 removes a negative charge that blocks the ATP from the binding site. As you can see in this illustration by Nelson and Cox 2017, in phase 3, the cyclin CDK complex forms, but phosphorylated TYR15 blocks the ATP binding site by making it still inactive. So no cyclin present, the CDK would be inactive. Then the cyclin synthesis leads to its accumulation. On the third stage, the cyclin and CDK complex forms, but phosphorylated TYR15 blocks the ATP binding site and then it's still inactive. Now, as a result, right here on 4, both of these mechanisms activate the cyclin CDK in many folds. Subsequently, the CDK phosphorylates the enzyme PTP protein tyrosine phosphatase, resulting in more CDKs right here in stage 5. This enzyme that dephosphorylates P-tyrosine 15 is a substrate for the CDK activated by phosphorylation. Ultimately, these mechanisms allow phosphorylation of proteins that targets the critical progression of the cell cycle. For instance, Nelson and Cox 2017 explained that even a break in a single-strand DNA prompts an arrest in G2 phase through activating the ATM and ATR. Now, ATM and ATR are serine threonine protein kinase, which are proteins that initiate cascade of response that inactivates the CDK. When this inactivation happens, the cell cannot divide until the DNA is repaired and another problems are fixed. Now, Nelson and Cox also described the degradation of cyclin subunit as highly specific and precisely timed, occurring all throughout the cycle. Initially, the mitotic process activates cyclin A and B and then destroy it to activate the CDK in mitosis. The destruction box recognition protein, or the DBRP, which contains a sequence of 9 amino acids, including arginine, threonine, alanine, and so on and so forth, is located at the amino terminus of these cyclins. The DBRP functions for recognizing this sequence to combine the cyclin and the ubiquitin, which is another protein by the enzyme ubiquitin ligase. This mechanism signals the enzyme complex proteasome for the process of cyclin degradation. If you take a look at phase 4, the increase right here, the increased CDK here would eventually lead to cyclin proteolysis on step 8 or in stage 8. This is because the cyclin CDK activity phosphorylates and activates the DBRP as well, causing proteolysis. Hence, if the cyclin level is low, then the CDK activity declines and slows down the activity of DBRP to complete inactivation through the DBRP phosphatase. Now, another mechanism that contributes to changing the CDK activity is some of the developmental triggers and cell division, such as the growth factors and cytokines. These two signals create the central signaling pathways MAPK cascade that activates the nuclear transcription factors JUN and FOS. In turn, these factors synthesize genes, proteins like cyclins and CDKs, and the transcription factor E2F, which enables the simulation of essential enzymes so the CDK and cyclin can enter the S phase. The last one is the action of the specific CDK inhibiting proteins. Nelson and Cox 2017 elaborated that the regulation of CDK in the cell cycle can be explained by analyzing the effect of CDKs on the structure of lamin and the activity of retinoblastoma protein. The lamin is a protein that makes up the filaments of the nuclear envelopes, which depolarizes and breaks down in mitosis due to phosphorylation by a CDK. On the other hand, the protein myosin, which is usually for muscle contraction, also acts in dividing the cells and cytokinesis, is also phosphorylated by CDK. The phosphorylated subunit of myosin disassociates the myosin from actin and fundamentally in active contraction to stop the splitting of the cell into equal halves. The dephosphorylation will reassemble the contraction machinery to prepare for the cytokinesis again. Finally, the retinoblastoma protein, or PRB, contributes during an arrest in the G1 phase. If there are damages in the DNA, its mechanism is bind to E2F, which is a transition factor that prevents the transcription of different genes such as DNA polymerase, which is necessary for DNA synthesis, this allowing the cell cycle to continue to the S phase. The event changes when cyclin E-CDK2 phosphorylates PRB and signals the continuation of the cell division. Furthermore, I mentioned earlier about the protein kinases ATM and ATR, which detects damage in DNA. The damage may be intrinsic, such as intermediates of metabolism, attrition of telomeres, oncogene overexpression, and DNA replication errors. Conversely, extrinsic damages are caused by either sunlight, carcinogens, ionizing radiation, or other anti-cancer therapeutics. That's according to Barnum and O'Connell 2014. The ATM and ATR phosphorylate p53, which is a critical component of DNA damage checkpoints to stimulate the synthesis of the protein 21. When there is a checkpoint dysfunction, it can either result from cell death or cell programming. The p53 can direct cells to apoptosis or senescence, losing the G1 checkpoint. Consequently, protein 21 inhibits the protein kinase activity cyclin E-CDK2, according to Nelson and Cox. It also keeps the PRB unphosphorylated and bound to E2F in the arrest in G1 phase, which provides the cell the time for the DNA repair before continuing to the S phase. This occurrence avoids the transferring of defective genes to the daughter cells. However, if the damage is no longer repairable, this mechanism also allows apoptosis, cell death, thereby preventing potential cancer development. Now, this article, The Development of Protein Kinase Inhibitors for Cancer Treatment, is from Nelson and Cox. I will discuss it briefly before I summarize the entire presentation. So the first one, Nelson and Cox presented an article, The Development of Protein Kinase Inhibitors for Cancer Treatment. The article explains that cells that are not regulated lead to massive production of cells that adversely affect the bodily functions as a whole. Cancer remains to be one of the leading causes of death, is caused by uncontrolled and unregulated cell cycle that creates defects in genes. According to Collins and Pavlitich, the first genetic alterations that showed contribution to cancer development were gain of function mutations. Mutations in genes may produce oncogenes that send signals for active cell division, rather than a normal rate cell cycle from the normal cellular proto-oncogenes, which promotes cell proliferation. Meanwhile, the tumor suppressor genes may mutate and produce proteins that do not encode proteins that restrain cell division, eventually resulting in the formation of tumors. Accordingly, the specific inhibitors of protein kinases may be an effective treatment of cancer, since these genes encode protein kinases that inhibit tumor growth and development. Some examples of these cases are the receptor tyrosine kinase, which shows that a mutation in gene for the receptor HER2 yields an RTK with activity increase up to 100-fold in approximately 30% of women with invasive breast cancer. The vascular endothelial growth factor receptor, or VEGFR, is another RTK that is crucial in the formation of new blood cells, and inhibiting this RTK might eliminate the food and blood supply of the tumor. Additionally, mutation of non-receptor tyrosine kinases can also signal unregulated cell division, such as oncogene ABL associated with a rare blood disease called acute myeloid leukemia. Specific protein kinase inhibitors might also be valuable as chemotherapeutic agents for treating diseases. Many drugs have already been studied showing positive results, and some of these drugs are as follows. Imatinib mesylate or Gleevec is almost 100% effective for patients with early-stage chronic myeloid leukemia. Erlotinib or Tarceva is effective against advanced non-small cell lung cancer by targeting EGFR or epidermal growth factor receptor. Sunitinib, the sunitinib or Nexavar and sorafenib Nexavar target protein kinases including vascular endothelial growth factor receptor and platelet-derived growth factor receptor to treat patients with gastrointestinal stromal tumors and advanced renal cell carcinoma respectively. Trastuzumab or Herceptin, cetuximab, Erbitux, and Avastin. These are monoclonal antibodies that target HER2, EGFR, and VEGFR for treating different types of cancer. Finally, to summarize it quickly, the cell undergoes cell cycle to divide, differentiate, or die. The CDKs are enzymes which are essentially important to cell cycle regulators. Their function is inhibited through the process of phosphorylation. It is always present in the cell but is only activated when they bind to cyclins. Some proteins activate CDK cycling complexes to stimulate the cell cycle progression, whereas other proteins inactivate the CDK cyclins to inhibit the cell cycle. Mutations in gene coding proteins may lead to formation of tumors and cause different types of proteins. Hence, as a finale, I will show you this video that shows a common mutation in a gene called APC, which often cause colon cancer in patients. It's a sulfate gene, APC. So as you can see, the ones that change the colors are the ones that mutated. Since they mutated, they will have difficulty to pass the next stage and eventually pile up, which becomes the tumors. So those are the information are the concepts in my presentation. And thank you for listening and watching. The references is here. And I hope you find my presentation interesting, and thank you.